Hello, everyone, and welcome to today's keynote webinar titled Commercialization and Entrepreneurial Lessons from Microfluidic Biotechnology Startups. This webinar is part of the 6th Annual Laboratory Automation Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of this presentation. Now, if you have any trouble seeing or hearing this presentation, just click on that help desk located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located in the abstract window below the presentation window and follow the process to obtain your credits. Without further ado, I now present today's speaker, Dr. Kalyan Handique, Vice President at Advanced Technologies at BioRad. For the complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Handique, welcome. You may now begin your presentation, sir.
Thank you, Susie. Good morning, good afternoon, or good evening, depending on where you are. Thank you for joining this webinar, and then thanks to LabRoots for allowing me to present this keynote speech. Again, the topic of my talk today is really going through the commercialization and the lessons learned through two of the biotech startups, HandiLab and Celsi. Please, type as many questions as you can. Try to answer as best as I can during the 1 hour talk and Q&A session. If need be, I will have Susie collate those questions, and then we'll see if we can. The ones that I'm not able to answer because of time, we'll try to get to them, later somehow. So again, I am based in Ann Arbor, Michigan. And then, before I go through my startup journey, just to give you a little bit of background on my career journey. So I got my PhD in chemical engineering at University of Michigan, 95 to 2000. I was fortunate to have worked in a highly interdisciplinary team. I was part of the chemical engineering, but I worked with electrical engineers and human genetics department. So from the get-go, I got a very, very exciting interdisciplinary environment, and we had really a nice team environment, trying to build something very, very unique. And as a result of some of the collaborative work at Michigan, we were we built a DNA analysis chip. I'll go through it later in the beginning of my talk about HandiLab. After my PhD, I started a company called HandiLab. It focused on automated DNA analyzers for hospitals, primarily focused on infectious disease. The company was acquired by BD in 2009, and then I stayed on with BD for about a year and a half. After that, I joined another startup in Ann Arbor. It used to be called De Novo Sciences, and it changed the name to Celsi. I joined that company in 2011, and then the company got acquired by BioRad in April of 2020. I'm still with BioRad. So between the two startups, it has taken up most of my last 22 years of my career, and I had an exciting time really starting these two companies, going through the entire process of ups and downs of this journey of taking a product concept all the way to products that are commercialized and that really made an impact to the either the clinical diagnostics market or the life science market. Let me start with a little bit of story about HandiLab. Again, I will try to narrate it from how I saw it. Different people that went through the company would have a different experience, but this is purely how I saw it, having started it right from the beginning and stayed through till the end. So how did HandiLab start? So as I mentioned, I was at University of Michigan, a PhD student, got a very exciting team to work on a DNA sequencing chip. So we were fortunate to have the right environment to build something very unique. So we created a DNA analysis chip in 1998. It was one of the breakthroughs in that year. So it was the first chip that could take small amounts of DNA, mix it with different enzymes, go through different enzymatic reactions, whether restriction digestion or some amplification, and load a microgel electrophoretic channel, go through the separation of the DNA bands, and then detect all the product in the same chip. It's a highly integrated chip that allowed a lot of things to be done from DNA in to results out. So it was pretty unique, and that really put University of Michigan, the group that I'm working on, in the map in the scientific community. In fact, this chip is in the Smithsonian right now. So it was very exciting to work on that. But having worked on a very fast-paced research kind of environment, I really wanted this kind of work to really get to the customer, make it into a product that people can use and say, hey, this is really beneficial for our work. And that kind of prompted me to really look into the startup area, because when I was doing my PhD, I often saw the best work was always coming out of the startups. And long story short, I got connected to some local investors who really were interested in helping commercialize this technology out of University of Michigan. And then I reached out back to my group, and I had another colleague in the PhD team, Sundaresh Brahmasandra. So we teamed up, and then we started HandiLab in 2000. And the vision was really a handheld laboratory. It's a handheld device that you would have a disposable cartridge that can analyze DNA. And then through the tech transfer, we got access to a couple of patents, mostly related to microvalve technology. In fact, it is the same thing that I worked on my PhD thesis, so I was pretty familiar with it. So it looked pretty good, and then we had this vision, and we were able to get excitement among the investors locally, and they put in money. But little did we know that it's not an easy ride. What we did at the university, and when we started looking into what really needed to be done to pick the right application, it took us a while. And then it also figuring out how to tailor this technology to a particular application. So we started looking into the infectious disease, because you had the need for speed. And then we soon realized that our technology was not complete. I mean, we could analyze DNA, get results, and then when we really started digging into details of how to apply it to a specific clinical sample, in this first case, we picked up something related to group B strep detection in pregnant mom, and we tried to really figure out how to do the sample prep. It took us additional development on the sample prep technology. The other thing that we did was, when we took the technology from University of Michigan and really started digging into the cost analysis, how much it would cost to build a complete disposable, we realized that the microfabricated glass silicon process that we had, done at university was not going to meet the cost needs. So we had to pivot to injection molded, technology. We also move the sensors, the heaters, and all the things that are required for the actuation, move it from the disposable to the instrument. And then I will talk more about microvalves later on. We had used wax as a way to move a plug of wax to block channels. We adopted nanoliter solder dispensing for the wax valve creation. We also had to create a lot of sample prep technologies to kind of meet the needs of the clinical sample. And then also we had to lyophilize the reagents, whether for the PCR or the sample prep, and also figure out how to store liquid reagents in the cartridge in blisters. So long story short, it took us another 5 years to really put it all together. So we did build it a complete integrated cartridge that had liquid reagents, dried reagents, and then it had all the fluidic network to connect all the different components required for sample prep to assay prep and then real-time PCR along with the controls. We moved all the critical sensors to the instrument, and then it's a really nice embodiment of a lab on a chip technology that we all dreamt about. And then that was not easy. It took us another 20 million over that 5 years. Luckily we kept innovating through the process, so we were able to secure funding from the venture capitalists along the way. But it did come with a lot of board changes, a lot of leadership changes. But the technical team had the tenacity to really pull it off. And then it really made we're proud of these achievements. By I think it was about 2006, we really had a true lab on a chip, and we started to focus on the clinical trial for the first application. We met with FDA. We had our preclinical testing, and we had lined up a couple of hospitals to do the clinical sample testing and compare it to other methods. So there I'll just go through some of the details. The lab on a chip cartridge had large sample chamber where you can put in a swab specimen up to about 1 milliliters. You could mix reagents in it. You could heat it up to the enzymatic lysis. And then it also had the nucleic acid capture magnetic beads, or in this case, in this embodiment, we use a certain size particles with the affinity for nucleic acid. And you could concentrate all these beads into a very small inline filter, just about 2 millimeter diameter. And then when you wash through all the junk and then you elute the nucleic acid, you could concentrate them down to 1 microliters, starting out from a 1 ml sample. So huge level of concentration, and you're also removing all the junk. And then you would have storage for the waste that comes out of it. So any lab on a chip operation, you need a lot of okay. So you need a lot of fluidic steps to be able to meter nanoliter amounts of reagents, mix it with the say nucleic acid DNA eluate. So you need specific times you want to do this in a microfluidic network. So we used wax that melts at a precise temperature. Those are preloaded in certain parts of the fluidic network, and you could open it up by using a very specifically designed microheater. And then you could also use these pumps that are part of the network with materials in it that expands to generate pressure, to create the pressure to move fluids around. So again, very clever utilization of different materials with the fluidic network to make this work through a instrument that had the sensors and actuators built onto it. We could also close fluidic channels when we need to block off parts of the fluidic network, either for some additional operation or for doing high temperature thermocycling protocols. Again, we use wax as a way to be melted locally and then heat trapped air pocket behind the wax to move the molten melted wax and close off channel when needed. And then for the liquid that needs to be delivered at specific times, we created these blisters with a piercer integrated, and then something in the instrument will push the blister down. The piercer will pierce a foil below. The liquid present in the blister, will just flow through the fluidic network and deliver it to wherever is needed. So it's all came together in this small box. It's almost like a shoe box size. Again, it was not a handheld that we wanted. It need not be, because that particular result, sample to result took almost an hour. And then you basically have a cartridge. You just load the sample, put it in the instrument, and close it, and then you get a result in an hour. So again, it's a nice, this is, you have to think about it, this is 2006. It's pretty much, we were able to achieve something that we all wanted, put sample in and get results in a certain amount of time. But it was good. But if you think about it, this was in 2007, it was still not good enough for what we needed to do. So the cost of the cartridge was still high. There were multiple layers, and then we would have needed additional R&D resource and design time to be able to take out costs. So I would say that exercise would have taken another year and a half, or maybe 2 years at least. And the instrument could run only one sample at a time. So in a 8-hour shift, you can do 8 samples. And then if you look at the cartridge manufacturing, there was a lot of different components integrated. So the QC burden on manufacturing was also high. Those could have been reduced over time if you did another design iteration. But even though we made the product, we probably had, in the final design iteration, we had over thousands of tests run, either mocked clinical samples or clinical samples. So it was definitely is going to the rigor to make sure it worked like it intended. But we still decided to stall that product. That was in 2007. So we had a working point of care product, but if you think about it, the market was still not accepting point of care molecular tests at that time. It is not the case right now in 2022. I mean, if you see, there is lots of point of care PCR tests for COVID or other respiratory virus right now in the market. Again, the other thing that we probably needed to at that time was reduce the time from that 1 hour down to below half an hour. Maybe it would have accepted, by the market. But if you look back, at that time, almost 100% of the nucleic acid testing was done in large reference labs or big hospitals. So the market had still not moved to the hospitals or other testing sites. So we really thought that we really need to focus the product to meet the customer need at that point of time. And the customer that we really started to target was the hospitals that were trying to adopt molecular testing. So we had to rethink how to change the product to lower the cost of goods, make it higher throughput, and also make it more open. Instead of running a fixed test, we wanted to give the flexibility to the lab hospitals in the hospitals to run their own PCR tests. And to do this, we had to abandon microfluidics for the sample prep and embrace robotics, because that gave us the cost advantage for reducing the cost and also improving the time. So, think about that. We've already burned through about 30 million in funding over 6 years. And we went back to the board and the investors saying, hey, sorry, our product didn't work. I mean, it's not going to generate the, revenues in the marketplace. So, we need to reconfigure. So, it was a do or die situation for the company. And then we got one last chance to get it right. And that's what we did the next 18 months. We built a product. We used to call it Jaguar at that time. We launched it in 2008. And surprisingly, just like the COVID pandemic now, in 2008, the winter, there was a H1N1 pandemic. And when we built this product and we started going out to the trade shows and other yeah, conferences, it really started getting picked up by the hospitals that really wanted to bring in testing for H1N1 flu and other things into the hospitals. And this was perfect because the instrument could run 24 samples at the same time. It will give you results in between 2 to 3 hours. And then it also had the flexibility to run a variety of RNA assays or other DNA assays as we were building the first few closed FDA assays on the system. So this was also we kept the PCR in microfluidics, and the sample prep was unitized in a reagent cartridge so that everything was pre-stored, liquid reagents, lyophilized pellets, as well as the tips required for running the instrument. So long story short, the we really started selling this instrument like hotcake, and a lot of companies wanted to partner with us, including BD Diagnostics. They were looking at putting hospital-acquired infection on a similar portable benchtop instrument, and really liked the form fit and the performance. So and the BD acquired the company in 2009, and then since then BD has done a tremendous job of commercializing this over the last 12 years. It has placed thousands and thousands of instruments and tens of millions of cartridges. In fact, when COVID really pandemic hit in 2020, and when testing was for COVID was especially related to PCR was becoming point at center, BD Max was one of the first to automate COVID testing. So and it has served the world very well with this instrument and the assays that can be run on this system. So again, I'd like to summarize with one slide about the journey of HandiLab. Again, it took us almost 10 years from starting the company to getting to a point where it got acquired. It was not easy. We probably took the first 4 or 5 years just to build up the technology required to satisfy the key requirements for the product. And then we along the way we are lucky to have got a variety of funding, either from VCs or Department of Defense, or ATP grant and things like that. But we really had to also pick up the right people along the way. We also hired a CEO from the diagnostic industry in 2004 that really helped focus the company into the right product. We built a point of care product between 2004 through 2006. Again, it worked, but still didn't meet the needs of the market. And then we had to pivot the company to a more benchtop higher throughput system, which is now called the BD Max. And that really was the right product that the market needed. And then the rest was kind of history. So again, I think getting that right mix and pivoting at the right time, I think is very key for us, to be successful. So if I took the lessons from HandiLab, these are the things that I would summarize. The key ingredients for a startup success. I think the team and culture. The initial team sets the culture of innovation. You have to really innovate against the incumbents and the other competitors in the space to build value, create IP. But as you are building the product, you also have to bring in commercial focused team to to build the right product and also grow the company. The focus is also important. As a platform company, you could go into many different directions. DNA analysis could be used in infectious disease, pharmacogenomics, veterinary testing. How do we make sure you're always focused on the right application or set of applications to get the company focused, because you are startups do not have infinite resources. There's a set amount of funding and time horizon for you to get it right. And then the right, you have to build the right product solving an unmet need at that time, getting that juxtaposition of market, product and the channel fit. I think is very important. And then you may have to pivot along the way to get to the right product, and not having the company explode. So that's very key. And then you do for molecular diagnostics, clinical diagnostics, because of the FDA requirement, the quality requirement, the rigor in testing. You really have lots of capital need. And then that also you need that capital to build the patent estate to protect the company along the way. You need patience. It's not gonna come easy. I've done 2 startups, each took away 10 years of my life each time. So you need the patience. And then again, timing is key. Getting the alignment of the right product, the right market conditions, and also having a channel to get it out to the marketplace. Okay, I'm gonna switch my focus to the next startup, Celsi. This was a totally different story. With Handy Lab, we had technology that came out of University of Michigan that was, being developed for over 5-6 years before that. But it's a completely different story for Celsi. So Celsi used to be called De Novo Sciences. It was founded in 2010 by 3 rookie biologists. Priya Gogoi, she's a molecular biology student. Saida, she's also a molecular biologist, and Chris Simmer also had a biotechnology degree. And they either met at school in the master's program or met at another startup. Chris and Priya met at another drug discovery startup in Ann Arbor. And then the company was focused on non-invasive cancer diagnostics for a variety of reasons. And I was fortunate to have mentored the team. It took almost a year to figure out the right business plan where they could apply their knowledge in biology in an area of unmet need. So they actually won the business plan in 2010 Great Lakes Entrepreneurs Quest, only $5,000, but that gave them enough confidence that they got the business plan correct, so they can really start putting together the technology around the business plan. So I joined the company in 2011. Again, it's a new company with lots of risk. There is they did have a good business plan. They have identified all the nuances. There were a lot of things that need to be developed on the technology, raise money, build product. So from my point of view, I was taking a huge risk, but at the same time I said, okay, let's give it a shot. For if I'm able to raise money around the business plan, we will keep going at it. So I joined in August 2011, and within a few months we were able to secure $500,000 by winning the world's largest business plan competition. It used to be called Excellent Michigan. So the first prize was $500,000. So we're able to really garner that money, and that really helped fundraise for the company. I was able to secure angel investors from investment for the company, and then we really started executing on our technology and our plan based on get after we got the money. So initially it was do whatever it takes to generate data. We created a microfluidic chip for cancer cell isolation from blood, and you can see there's a slide, we call it de novo chip, below a microscope, and then we had tubing going in to run blood through it. We used to call it internally Mr. Jankity, but it gave us enough data to keep iterating on the product that we were planning to build. Long story short, it took us another 2 years to have a minimal viable product. We had a chip, we had a system that could run the microfluidic chip with blood, and here we are focused on circulating tumor cell, isolation, and then downstream analysis. And we also build a platform to be able to do all the different staining and other post processing. And then we launched the product at AACR, a conference. This was in DC, and you can see the early team. And it was, because of my experience with Handy Lab, we wanted to make sure we were, no matter what, we're always in sync with the market. So we kept going to the conferences to get feedback, make sure we're always designing the right product. So in over the next few years, we generated a lot of data in partnerships with variety of cancer institutes and other biotech companies. So we had this particular microfluidic chip. We call it Cell Select. So it's almost like a 1 inch by 3 inch slide with a fluidic network that could process whole blood directly, and you could run blood at the rate of 1 milliliter per minute, and have, even though it's a fast flow, it was able to capture the cancer cells with very high efficiency, capturing them in their own chamber, and then have, if you need, you could remove the cells out for downstream culturing or molecular analysis, and or you could also analyze the cells in the slide using different antibody stains. So we really allowed very rare circulating tumor cells from blood to be enriched, and then it was very, very promising technology. And we also started, we met with FDA, figured out the what the clinical trial would look like, but that needed tens of millions of dollars and another few years to go through the clinical validation, because you have to go cancer by cancer, whether it's breast or prostate or things like that. And then you have to go through the regulatory approval. So it needed a lot of funding for taking it through the paces. So, and as I tried raising money for this in a product, this was in 2015, 2016. There was it was very difficult to raise money for another circulating tumor cell company, even though it had tremendous advantages in cancer prognosis, monitoring, and therapy selection. The funding was very tough at the time. There was a previous device, FDA approved device called Cell Search, that was already in the market, but it had poor performance. It missed circulating tumor cells because of the exact antibody cocktail it used for capturing, and that really stunk up the market. And then in parallel, there were a variety of liquid biopsy companies based on cell-free DNA enrichment that was raising billions of dollars, the Gardens, the Grails, the Foundation Medicine, and things like that. So that really sucked up the money. For it was not much money funding left for CTC, and we really had to figure out what to do with the company. So we went back to the drawing board. We had IP that we had filed early in 2011 on other single cell analysis. So we really pulled up other IP and technologies we had in our in the back burner, and we started focusing on a new area which is more in the research market, how to uh, capture hundreds of thousands of cells in viable format at the same time, and allow an instrument to be able to do a variety of analysis on them. And essentially it's a research market called single cell analysis. We're trying to parse out data from cell by cell level and looks at millions of cells at the same time. So we realized this was a very rapidly growing market, but it needed you to be active very quickly, and deliver a product. So this is what we did. It's a Genesis instrument. And then over the next 2 years, 2018 and 19, we really had to focus on the single cell applications. We invested lots of dollars in developing protocols for single cell capture, single cell sequencing, with highlighting our increased cell throughput, increased cell viability, and very streamlined library preparation steps. We increased the breadth of our patent filings in single cell based on our early priority date for our early application. So I remember in between 2018-19, we probably had 20 new applications that we filed. We really beefed up our patent estate, and then we also brought in a really top-notch commercial team from California into Ann Arbor. These are folks that understood the sequencing market and also the single cell analysis market, having worked at other single cell companies like Fluidigm. John Stark became the CEO. I actually stepped down from that CEO role. Pat Cook on operations, Chad Alexander on sales, and David Cohen marketing. Again, we really needed to focus on commercializing very quickly on our enabling technology. Long story short, in 2020 we got acquired. Actually, we got acquired on April 1st, 2020 by BioRad, the day the stock market had crashed and the pandemic was raising. We're very happy that BioRad understood the value of Celsi and now are integrating the Celsi technology into variety of product lines for BioRad. So again, just like with HandiLab, very similar kind of story. You slightly different start. The company started out with an actual business plan, with a desire to go into a certain area. It was able to leverage the business plan to garner not only a business plan money but also angel money. And then we focused on the circulating tumor cell technology development for the next few years, and then started generating lots of data. And then we did run into funding issues for funding that CTC program. So we had to pivot the company into microwave based single cell analysis application. Luckily, we had really good IP that really allowed us to keep focusing on the new area, build momentum with investors, and also able to get in a commercial team. And then as we gathered traction in the market on the single cell analysis, we eventually got acquired by BioRad. So if I want to summarize the lessons from Celsi, again, you need a team. With Celsi, mentoring and nurturing young entrepreneurs can create great unexpected results, because it the name of the game is resource efficiency. You want to get as much as you get done, and in this case, we had 3 young, hungry entrepreneurs that really wanted to create a difference, and that really helped seed the company and its plan without spending too much money early on. The initial team's commitment and passion really sets the innovative culture. That's what really builds the competitive advantage, the technology that really attracts dollar and other folks to join the team. But you do need a commercial focused team eventually to grow and exit. You need lots of funding. Startup savvy and pragmatic investors with deep pockets are needed to weather the long road. This is gonna take 5 to 10 years, and then you need the access to capital to be able to do what you need to do. And then the pivots, the changes are difficult but very essential to get the right fit of product, market, and channel. Again, that's the same team that emerged from HandiLab. You may be building something very cool, but the market may not be right at the time. So getting that right, being at the right place at the right time is important. Some people call it luck, but I think it is. If you're not in the game understanding, you will not see the luck come by. And then most important thing, the right product, You have to build the right product, solving an unmet need that customers want to buy right now. I think that's important. And in the case of Celsi, the Celsi's innovative patent estate in microfluidic microwell single cell technology was the key in its successful exit. So that's all on my talk. Now I would be open to any questions that you might have.
Well, thank you very much, Dr. Handique, and for that informative presentation. And we will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Our first question says, Dr. Handique, thank you for such an enlightening story and of your entrepreneurial journey. It amazes me in both of your companies required a pivot, which must have been a gut-wrenching decision. Business advisors often tell you to stay focused on an area and not to dig multiple holes, yet say pivoting was necessary for success. What is the process, intuition, or even criteria that you use to make the decision to pivot? And what advice would you give to those who must pivot?
Okay. So for, I'm coming from a technology point of view. When you are creating a company, you really want to create more technologies that you encompass into a particular product so that you have options down the road. And then it's important, especially in this biotech space, to create more IP at the beginning, even though you may not be taking it all the way through, the development process into a product. So I think it is very important to be singularly focused at any one time on a particular product and a particular customer segment. And you have to get alignment of the entire team, your board, the business advisors on the same singular focus. But as an entrepreneur leader, you always have to keep looking at the cues from the market. If the market is not buying, means you are not designing the right product, or you're not at the right time. I think you have to keep getting that feedback. And then I think you will sense it that, hey, at some point, if we are trying to raise money on a particular concept for a few years and haven't got it, that's a good enough feedback that, hey, you need to change. And at that point, it's a difficult process of getting the whole company aligned on the change, what the change is, and also the board and the business advisors on the decision to change. It's a difficult process, but if you get the data to everybody that, hey, we've been trying and this is not working out, we need to change. I think it's important to be able to do that. I think you have to be, you have to do honest job all along the way, and very, very honest with the feedback and also with the board. I think eventually you can do it. It's very difficult, but I think if there is honesty on all sides, you are able to do that. You're muted, Susie.
I apologize. Thank you so much, Dr. Handique. We have time for a couple more questions. How important would it be, the location for the biotech startup?
Yeah, so for me, both the startups were in Ann Arbor, Michigan. When we started Handy Lab in 2000, Ann Arbor was not known for startup. I think because of the long time, because we got the money from local investors, and technology needed incubation for many, many years. I think it was good that we stayed in Ann Arbor, because we were able to grow the team and grow together for many, many years. And that was what created tremendous intellectual prowess for the team. And I think it helped us to stay in Ann Arbor. We had opportunities because of funding to move to the Bay Area, but it didn't pan out. So I think it was the right thing for Handy Lab. And then over time, Ann Arbor morphed into a much more entrepreneurial. So, Celsi was definitely the right spot to be in Ann Arbor. So I had some good viewpoints. I mean, if you can, I think the location is important. You have to be in one of the startup hubs, whether it's the Bay Area or the East Coast. But you also have to be cognizant of the fact that if you go to California, if your technology is really not very, if it's not enticing, it is not sexy enough, it's hard to keep your people intact because there is so many opportunities for talent. So in a if but in Ann Arbor, I don't think that mercenary attitude has come to the tech people yet. So I think it depends on what you want to accomplish. If you want to keep the good people intact for many years, maybe not, California may not be the best option unless you can really keep them around. So the location is important, but it need not be at the east or the west coast to really be successful. That's my whole point. It depends on exactly what stays and what field of use you are at.
Thank you so much, Dr. Handique. And we have time for one more question. What are you currently working on at your current job at BioRad?
Okay, so that's a good question. So BioRad acquired Celsi through the life science division. So after acquisition, actually, I had an interest in going back to the clinical world because I really uh, you know uh, like the impact we can create in the lives of people through innovations in clinical diagnostics. So I actually moved to the clinical diagnostics group of BioRad, and BioRad has tremendous amount of technology in PCR, digital PCR, and variety of cutting edge molecular techniques, but it never had a focus from a clinical side. So I'm actually helping innovate on a new platform for molecular diagnostics for BioRad. And then besides that, I get involved with lots of M&A activities to really grow the molecular business for BioRad. So we're building a small team in Ann Arbor while working with my boss in California to do whatever it takes to build up that molecular presence in diagnostics for BioRad.
Thank you so much. And you know what? We actually have time for this last one that just came in. Is the development agreement a key step in getting acquired?
No, I mean you could. In the case of HandyLab, we did have a development agreement with BD for certain menu, and then I think that accelerated to us being HandyLab being acquired. It could be one of the steps, but we with the second company, Celsi getting acquired by BioRad, we never had any development agreement. It was just we went through a process where we had many companies look at Celsi, and then BioRad essentially won out in the end without any development agreement.
Dr. Handique, I want to thank you for your presentation today. Would you like to provide any final comments to our audience before we go?
No, I think yes. One comment I would say that the area of biotech, especially bringing new innovations, is very hard, but it is extremely important for this world. We've seen in the last 2 years, how diagnostics have really helped manage patients better, and I think the importance of getting diagnostics, cutting edge diagnostics, is important. When you lose near and dear ones, it will hit home. I think the researchers that's in this space have to continually innovate to get better and better tools and tests into the market, because you will get a chance to impact this world tremendously more than anything else than in any other fields in this world.
Well, I want to thank you again, Dr. Handique, for your time and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted on demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until May 18th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, take care, everyone. Bye-bye.
Thank you.